we actually use the same scientific advisory board, which meets every spring in San Diego. And the scientists who have been doing the research come and present their findings. Other scientists who are applying for a grant might be there as well to present what they might want to do the work on. And the model is working. Yes?
So it really is a snowball effect, because what you're doing is really venture capital money. So you give a young researcher or researcher $50,000 or $100,000, and they take that money and they can actually do some research and get some preliminary data. Once they have preliminary data, that's when they can apply for NIH funding. So it really is leverage.
Yeah, you're right about that.
It's exactly, you don't need a lot of money, but if you get $100,000 to get some preliminary data, hire a grad student, then that's when you can apply for NIH dollars. And that's why this is, that little chunk of money is...
I'll show you a perfect example of that when I talk about a University of Texas Austin study that we're funding, okay? Where they're getting a little bit of money and they're getting a lot of good results from it, okay? And then that can, it leads to more support from bigger sources of dollars. Scientific Advisory Board. Here's Michael Harpold from Down Syndrome Research and Treatment Foundation. He and I talk a lot, okay? He's really the leading light in terms of directing a lot of the science you're going to see here, more directing the strategy itself, making sure that the right areas are being covered, okay? But you see Roger Reeves from Johns Hopkins, Linda Dell. I'll talk about his research. He's very engaged in identifying or developing cognitive test batteries so that when the clinicals come, there's an objective way to measure the effectiveness of drugs. Okay, Leslie is at University of Colorado. Ronald Evans is a renowned scientist out in California. Trust me, when the dollars are being granted to researchers, it's being under the direction of a very top-notch group. Here's the whole research strategy. Up front, you're talking about discovery research, where you're using models. In this case, I'll be talking about a mouse model. Try to start understanding what pathways might be contributing to the cognitive difficulties. Once you start getting downstream here, you start looking for potential drug targets, which I'll describe. If you find a good drug target, you start optimizing the drug that is directly against the target. You go through this pathway of preclinical development and an investigative new drug, 3-phase clinical trial, and then FDA review and approval. Drug target, I use that term and sometimes I'm like, what are you talking about? This is nothing uncommon. All a drug target is, it's a structure that undergoes an interaction with something on a membrane or a certain chemical or receptor on a membrane and makes something change, okay? So it might upregulate or downregulate cell interaction. So that's all a drug target is. And many of the drugs that we talk about, breast cancer, arthritis, Alzheimer's disease, there's a drug, there's a specific drug target, and then here's the action, okay? Not to spend too much time on this, but you can make the same statement then with the drugs that are starting to be identified as potential therapies in Down syndrome, directed against membranes of neuron cells, directed against receptors on the membranes, directed against or upregulating growth factors, that sort of a thing. There's 324 approved drugs that are specific biochemical targeted drugs that are approved by the FDA, so this is not uncommon. There's drug targets for ADHD and ADD and that sort of a thing, So when I describe drug targets, it's a specific approach to addressing a condition. I'm going to summarize the research, then we'll get a little bit detail of each of these bullet points here. 6 different pathways that hinder learning have been uncovered. There's going to be more, okay? Some may be more effective than others potentially in terms of a possible pathway for an effective drug, but the pathways I'll be describing, total 6 at this point, these would be targets for drug therapies. 2 involve the degeneration of nerve cell circuitry. This is kind of like the Alzheimer's condition that you see connected with Down syndrome is connected with Alzheimer's, a loss in function. In the case of as you probably hear amyloid plaques that get formed in the brain cells of persons with Alzheimer's disease. You see that same connection in Down syndrome. So a couple of drug targets are directed there. Two other pathways hinder learning are related to signaling between nerve cells. Okay? There's always receptors on cells, and neuroreceptors and neurochemicals that make nerve cells talk to each other. I'll be describing that. The very first clinical trial is going to be underway within a year by Roche on testing a drug like this. We just announced this on our website and had a big press release on this. It's exciting news when you think about it, isn't it? Because I think 7 years ago we were getting this. Roche, a well-respected world leader in pharmaceuticals, is starting a drug trial. There's also a pathway that's been identified that associates degeneration of specific brain cell circuits with contextual learning. Contextual learning is, it's tested in mice by their ability to form nests. Contextual learning in us is like how we're, let's say in a mall, we're trying to remember where Best Buy is, and you think, oh, that's right next to Sears. Okay, that's the type of learning we're describing here, really in the hypothalamus. And so Dr. Salehi in Stanford, who we're funding, is working on a drug that would offset this degeneration of brain cell specific neuron circuits. This is really exciting work here. This is the work being done by Roger Reeves at Hopkins. The impaired formation of nerve cells and nerve signaling during development, actually embryogenesis, the growth of the baby as a fetus, and even post-birth, the work he's doing in mice right now, and not that far from clinicals and human clinical trials is underway. So that's a growth factor related pathway that I'll be describing. Questions at all for any of this? I'm going to try to stay away too much from the technical side of things. If you don't understand anything, please raise your hand and I'll try to clear it up. We all know trisomy 21 in humans is the source of Down syndrome. When you have an extra chromosome, right, which is where all of our genes reside, in general we have 2 copies of each gene. In the case of Down syndrome, we have 3 copies of the genes on chromosome 21. So you have an imbalanced expression of the genes on chromosome 21. So if you have 3 instead of 2, you could do the math, like a 50% increase in the production of certain proteins tied to certain genes tied to the 3rd chromosome. So there's an increased expression of these genes. You also see not just increased expression of a protein, but as you know, genes, or may not know, but genes also have regulatory functions. If you have an extra set of genes, then you're seeing altered regulation of functions within neurocircuitry and so forth. Not all of the genes are likely to be dosage sensitive. What I mean by that is, in some cases, even though there's a 3rd extra gene, they don't see any difference. So the production is about the same. All right. So not all features are altered by gene copy number. Everybody understand that? It's pretty critical to what I'm talking about here. Because what I'll be talking about is that some of the pathways that we're seeing are caused by overexpression of certain things. Or downregulation. Why? Okay, you know, either because overexpression because there's an extra chromosome, so you see extra APP, which leads to the plaques. Okay, that's an example of that. You'll also see the altered regulations of genes leads to impaired nerve cell communication. All the work you're going to see from this point forward is generated by studies on a mouse, the TS65 DN mouse here. Okay, it's a mouse model of human chromosome 21. So they were actually scientists, and this was quite a bit ago too now, 6, 7, 8 years ago where this mouse was developed. So they were able to bioengineer a mouse that is as close to triplicate 21 as possible. There's actually 3 chromosomes in mice that have genes kind of homologous or equivalent to the chromosomes on chromosome 21. 16, 17, and 10. Chromosome 16, 17, and 10. 16 has approximately 112 genes, chromosome 16 on the mouse. 112 genes that very much correspond to a certain region of chromosome 21. So when the scientists developed their first mouse model, they concentrated on this building this TS65 DN mouse, which don't get too worry about this here, but here's this little segment of TS65DN chromosome compared to the human chromosome. But that's a pretty good chunk of genetic material that is trisomic in this engineered mouse. And these genes on here are equivalent to genes in the mouse, in the human condition, okay? So if you take this chromosome right here and blow it up like this, one section of it contains a number of genes, but there's some really, this really critical region, they call it, Down syndrome critical region of genes that are very much connected with conditions associated with cognitive difficulty in Down syndrome. I highlighted a couple, the APP amyloid precursor protein, the Alzheimer's related protein, okay. The DIRT1A is another very interesting gene that seems to regulate nerve cell communication. So it's a great model. It's the best model. Ideally, you could try to get a trisomic for all of the genes equivalent to chromosome 21, but they just haven't been able to do that. The mice they try to create aren't viable when they do that. Okay, but this has been a fantastic model to try to create a mammalian system to identify potential drug targets. Pretty much understandable to everybody here, what I'm describing, okay? So I'm going to go into some of the little bit more of the detail of the pathways. Neurological manifestations in Down syndrome. Here are the three main areas that are getting well understood now. Impairment during neurogenesis, during the development of the brain cells, leads to reduced cerebellar volume in TS65DN mouse. I'll show you a demonstration of that in a second, a little picture. The volume of the brain in mouse with Down syndrome is smaller than a control mouse. Inhibition exceeds excitation in nerve cell signal processing. Our brains are just firing all the time with neurotransmitters and receptors. You're exciting the signal, you're decreasing the signal is a real proper balance you need to have so that when I'm hearing something, I can process it. It goes to my short-term memory to my long-term memory, which leads to learning. It's a real critical balance, and if that's offset a little bit, then it creates the cognitive difficulties that children face. Okay? And not to belabor this again, APP gene overexpression and protein interference with instructions for neurons to thrive. Okay, so there's continual signals for growth, regeneration to a degree of brain cells, but when this APP gene overexpresses, you have a degeneration of specific neural cell circuits. A little more detail on this first one. Okay, this is the growth factor neurogenesis discussion, neurogenesis impairment, that first bullet point in the previous slide. So, during neurogenesis, you have this neural crest starting to form, really early stage of the development of the blastocyst and so forth as the fetus develops. And you have granular precursor cells that then become brain cells. And they become brain cells because there's a growth factor, and this is a Sonic Hedgehog. That was, my kids tell me that's a Sega game or something like that. And as Roger Reeves says, it was named by a scientist with a sense of humor. But so there's disruption of this Sonic Hedgehog signaling pathway leads to cellular abnormalities and a reduced volume. Okay, so this is P6, which is 6 days after the birth of a mouse. Here's a euploid cell, the proper number of chromosomes. Here's the trisomic to a degree TS65DN mouse, and you can see the difference in brain volumes here, caused by disruption of the signaling pathway, which prevents granular precursor cells to become brain cells, leading to this. Dr. Reeves' studies in the last couple of years are really exciting. He's treated TS65DN mice with this Sonic Hedgehog factor activator called SAG 1.1. Very exciting. This rescues this cellular abnormality. And this activator drug restores the development of the cerebellum and improves specific forms of learning and memory. And let's imagine that, okay? That not only is it demonstrated anatomically by a restoration of a more normal brain size, but it's also demonstrated by the studies they do on learning with the mice themselves. He is, and it's detailed in the summary of the research grants there, but he is not all that far from gathering enough information to sit down with the government and start trying to identify clinical sites to test this. Inhibition exceeds excitation in nerve cell signaling processing. Nerve cells respond to neurotransmitters, and it's a balance between excitation and inhibition. So you need this proper nerve cell transmission to have this proper balance. In these mice that I've been describing, the inhibitory signals are too strong. Okay? Yeah, you're supposed to have enough inhibition and enough excitation, but in this case, I'll be a little more detail, the inhibitory signals are too strong, leading to disrupted neural signaling in the brain, in the hippocampus region. So if we look at a couple of nerve cells here, okay, you have this axon. You guys remembering your biology, nerve cells, okay, the nucleus, and axon, and dendrites. And they're constantly communicating through neurotransmission, right? If you take this little section right here and blow it up, okay, so you have neurotransmitters, storage vessels. These neurotransmitters are released, okay, and then they are supposed to transfer over to the next nerve cell and be bound by a receptor, okay? If there's overproduction of something, in this case, what I'm going to call GABA A and GABA B, there's an overproduction of GABA A and GABA B, which are could be tied to the trisomic condition. If you have over-inhibition, as I represent there by this red arrow, okay? So the nerve cells are over-inhibited, and the communication is decreased. So we call that GABA A, GABA B mediated inhibition. These two GABA molecules here, neurotransmitters, are somehow impacted by the trisomic condition. It's not exactly known how yet. Okay, so when they're looking at this pathway, it's more to treat this as opposed to understanding exactly where's the source of this. But the data are very exciting here. So the Roche clinical, which I'll spend a little bit more time on in a second, is actually testing this pathway right now in phase 1, which is testing the safety of a drug that they have, okay, in a population of 18 to 33-year-olds, around 30 subjects. Really, really exciting. This is the drug. The drug that they are testing is related to what I'm describing here. So if you can add, in this case, a drug to go to this drug target, these GABA A, GABA B receptors, you can restore proper neural signaling, leading to improved memory and learning. So again, if we look at this little cartoon here, you look at here, here's the over-inhibited situation. But if you can come in here and block the GABA A receptor, which is causing the over-inhibition, you can upregulate then and restore more normal chemical balance in nerve cells. This is all being done in the mouse model. Again, this is what is beginning the Roche clinical trial. Y'all follow that then? Mm-hmm. Imagine that? So.
Can I ask a question about that medication? Is it a brand new medication?
It's a drug that they have. They call it a molecule. It's not a drug yet. It doesn't become a drug until it's a. But it's a molecule that.
Is it PTZ? Is it a form of that, or is it different?
It is a what?
That the drug, I think, is it PTZ? That
no, it's not PTZ. It's not phenylene triazole. No,
okay.
It's not. Okay, it's a version of that though, but it's not PTZ.
Okay.
So this was the work Stanford was.
This is the Garner's work.
Garner's
okay.
work.
Mobley, who's now not at Stanford any longer, but at UCSD.
Yeah.
Okay, so that's the work. I'm glad you pointed that out, right?
So what is the name of the drug?
I'm sorry?
What is the drug?
It's a number.
A number.
They give it a number.
Right.
I bet, if I knew the researchers and sat down, then I'd probably get it, but I'm just going off what I read in their press release. I can't remember. Okay.
Are these drugs that are being developed specifically for this purpose? Or
No, they put some of these. In this case, it's a drug that has been looked at in other areas as well, too, for other reasons. But once they understand what this pathway is, they say, hey, this might be a good GABA A receptor blocker. Okay.
Is it a drug or a chemical that's used now in people, or that's used for other?
No, not at the moment. No.
Neurological.
There are some cases in which drugs that might have, like PTZ, is actually used out in Asia for gosh,
Isn't it a
I think
cough syrup
hypertension or something.
Yeah, and a cough syrup.
You know, so it's approved.